Thanks. Good afternoon, everyone. My name is Wan Xingyang. I'm from University College London, and currently I'm a PhD student. Right, thank you. Today, I'm going to talk about how we can use terrestrial LiDAR for non-destructive biomass estimation. Right. Oops. Can we go to the previous slides, please? Okay, so biomass is defined as the dry mass for living or dead vegetation, and it is an important indicator for forest productivity and carbon stock. But in destructive measurements from biomass is resource intensive and requires heavy manual labor. As we can see those two videos here, can the video be playing? Can you please click on the videos to see if it can be played? Right, sorry about that. So those two videos just basically showing the process of felling a single small urban tree and then weigh the tree segments piece by piece. The whole process can take up for a few hours just for a single tree. And even with a professional team, let alone estimating biomass for a large tropical tree or across large scale. Allometric models is another option for non-destructive biomass estimation, but it could lead to high undensity when applying beyond valid species or diameter range. The launch of JEDI allows for above-ground biomass estimation for large scale, but it requires ground truth data for calibration and validation. So here comes in terrestrial laser scanning, or TLS for short, which provides the opportunity to deliver unbiased tree-level AGB estimation with the error independence of tree size. So this video cannot play. Right. Can you please try to click on the video? All right. So here is an example for the scanning pattern we use on our scanner, Regal VZ400i, and that's a animation of the 3D point clouds the scanner captured from around 20 scan positions. There is a trade-off between the scanning time and the accuracy. The pattern we use takes about 3 minutes per scan with the accuracy of 40 millimeters at a range of 20 meters away from the scanner. The measure rate is quite high, 250,000 points per second. So we can obtain fairly dense point clouds at the trunk and most of the upper crown. So if we can retrieve volume estimate from the point clouds, then we can estimate the biomass from by multiplying the volume and the basic wood density. There are a few methods that can already retrieve some basic geometric attributes from the point clouds by reconstructing 3D quantitative structure model, but the current methods do have limitations to do with selection of input parameters, reconstruction of the topology structure, as well as estimate the branch radius, especially for small branches. Take TreeQSM as an example. TreeQSM is a widely used method for TLS-based tree reconstruction. It shows good performance for volume estimation, but it requires to run hundreds of iterations for a single tree to select the optimal parameter combinations from the manually selected candidates. Besides that, the topological relationship of parent-child branch, as well as the radius estimate, are not necessarily correct, even if the volume estimation is quite close to the reference. As we can see in the figure, not working, okay. The lower left figure there, that's the estimation of total branch length, and we can see a clear underestimating trend. To overcome those limitations, especially gets better topology reconstruction, we proposed a graph-based method which do not need a user to give any input parameters, as well as can retrieve detailed morphological and topological traits at branch level. And the script can be deployed on HPC platform, which allows for plot scale reconstruction. And the figures above there shows the main workflow of our methods. So firstly, extract skeleton nodes from the point cloud, and then build a skeleton graph based on the skeleton nodes, which can provide a topological relationship of the individual branches, and then estimate the radius of each branch segments to generate a quantitative structure model from which we can deliver geometric attributes. We tested our method on 4 large tropical trees with destructive measurements. The data we use are from Andy Bird's 2021 paper. It provides the TLS data of these 4 large tropical trees as shown in the upper right figures there. And it also provides destructive measurements of wood density and above ground biomass. Compared with his manual measurements, the relative error of our modeled above ground biomass falls within 15%, while the estimates from pantropical allometric model can overestimate 30% of above ground biomass for a single tree. When evaluating our reconstructed model of tree number 2 and number 4, whose relative error is over 10%, I found that the major underestimation of tree number 2 appears at the lower irregular budgets there, the upper left figure there, and the magnification with large proportion of missing points. So the missing point will cause lower radius estimation and thus lower volume estimation. And for tree number 4, it overestimated twigs because of the remaining leaf points. For the other 3 trees with just woody points, our methods works quite well at upper crown. So we recommend to do leaf and wood material separation before doing the reconstruction. Since our methods can retrieve topological relationship of branch structure, so we can characterize tree attributes at branch level. So for example, here we plot above ground biomass distribution in branching level, in branching order. So branching order here determined by the vacation, which means the number of branching order add up by 1 whenever a parent branch split into multiple children branch. The bar graph underneath there shows that the majority of above ground biomass stored at the lower part of the tree, especially the stem. So for tree number 2, we can see that it has more than 50% of above ground biomass stored in the main stem. And we can also plot the above ground biomass distribution in branch diameter class. So here we classify the branch segments into 5 centimeter intervals classes, and then calculate the total biomass for each class. So what we found is that we cannot ignore the small branches. For example, the branch with diameter less than 10 centimeters dominates at the upper crown and can take quite a bit above ground biomass of the whole tree. The reason why the main stem radius is not gradually decreased is because the cross-section of the irregular budgets is far from a circle or ellipse. So when we fit a cylinder to those parts, it could have certain uncertainties. So we will try to find a better way to do this in the future work. One of the promising feature of our method is that it's able to segment individual branches with structural information. So here, for example, that's the example of the 10 longest branch extracted from the point clouds. And on the right, there's the separate plots of those branches. And then we can plot the radius change of the individual branch against the distance from base or against other attributes and explore the taper information. We can also explore the branchification distributions. So interestingly, these 4 trees are from 4 different species, but they have similar bifurcation patterns. Around 70% to 80% parents give birth to 2 children branch, and 14%, 21% parents have 3 children, and less than 6% branches have more than 3 children. To sum up, our method is a self-tuning parameter algorithm which can deliver accurate branch segmentations and fitting, and can give detailed morphological and topological traits at branch level. And the script is available for batch processing in an automatically way, so which are available for plot scale reconstruction. But we also have certain limitations. The branch radius estimation is affected by the point cloud quality. For example, the offset point clouds caused by the wind effect, which shown in the right figure there, can make the radius overestimate. In the opposite, the great level of occlusion can make the radius underestimate. And so we can maybe do a better job at fitting the branch tips and irregular bunches for the tropical trees. And for the future work, we will try to overcome those limitations as well as get more benchmark data to validate 3D reconstruction methods. So the pictures, it's a shame that the videos cannot be played, and it's showing the field work we are doing over the summer to get some validation data. And after that, we will combine it into a pipeline to automatically estimate above ground biomass and structure at plot level. And besides that, characterize tree architecture as well as explore tree structure and function. So lastly, a brief advertisement. We will hold a biannual conference, Silver Laser 2023, in September next year at UCL. The conference is focusing on laser scanning in forestry. So if you're interested in, welcome to follow us on Twitter, and looking forward to seeing you in next year. Thank you.
So thank you for a very excellent talk. Have you got any questions?
Hi there. I'm just wondering whether you could speak to how well the automatic extraction works if you had, say, epiphytic growth or lianas or vines growing in the trees as well.
Yeah, that's an issue. So we recognize all the points as woody points. So we have to do the separations before that. So if it contains lianas or other noisy points, the radius estimation would tend to overestimate.
Thank you. That was great, and I wish we could have seen the videos. I'm sure they were amazing. The trees I'm used to working with often have breaks in them, or the branches are snapped, or fire has got in. So they're very unusual shapes. And I wonder, with this method, could you understand how damaged the trees are because they no longer obey some of the scaling laws that you might expect because branches have been removed or damaged? And I wonder if that's something that could be examined with this kind of data set.
Yeah, that's an interesting thought. We doesn't input any assumption of the tree structures. We try to reconstruct the tree as what it looks like originally. So if there is a damage or many cuts on the branches, then it can reconstruct that shape.
Thank you, Wanjing, for a fascinating talk.
Thank you.
Yeah, I've got one question, and that is about the intensity of the signal. Is it possible perhaps to use the intensity of the laser signal to classify green vegetation like leaves and woody vegetation?
Yes, that's a totally additional topic, which a lot of researchers are trying to figure out how to get a better separation between leaf points and woody points. Intensity is one of the important information to separate them, and as well as we can use there are other methods, for example using graph-based information to see how the connection between the woody points and the graph the leaf points to separate them. Yeah, that's another topic.
That's really great. So thank you.
Cheers, thank you. Okay, the next talk is going to be given by Anna Maria from Manchester, and this one's concerned with the role of biomass in fire.
Thank you. And it's me managing it. Okay, so thank you very much. It's really nice to see so many known faces. This is kind of the methods. We still don't have like proper results to show, but like I also want to introduce the project, the UK National Fire Danger Rate System. So it is a project like to start building UK national, not to start, like to understand what is necessary to build a UK national fuel fire danger rate system. So I will give like a little bit of a background about what understanding the fire regimes are important in the UK, an introduction of the project, and also what are the methods that we are working on for building up like the UK national fuel map, and a few like questions if you have. The UK, it's routinely experienced wildfire, especially in the mid or late summer, and then in recent years it has been increased. Here we can see like by area, by burn area, and also like how it developed in the different years. This is like done with FIS information. So you can see in how 2022 had like large number of fires in the UK, especially, well, this is not related to this map. So, yeah, so like how it's important, and then these wildfire incidents require like lots of hours for fire and rescue services attendance. So here we have like a maximum daily fire water range index from 2000 to 2022. So we can see like the dates, those are like the dates in months, and we can see, for example, in here like the maximum risk of fire, like it was like in the heat wave of July. So like this is not fires, but this is like the risk probability of fires if an ignition occur. Also, like the UK has like a unusual complex regime that have like different, like traditional management that includes burning, and then it comes like it develops in large and small scale wildfires. So the largest number of wildfires occur in built-up areas and also like in the rural interfaces. So because of that, so this project was stopped just to see what was necessary to assess, manage, and mitigate the impact of those wildfires. So the idea is to establish and test the scientific underpinning key components to to build an effective and tailored UK fire danger rate system, and then included the impact of the current fires into this regime. So there is like the project team. So it is Manchester, Swansea University, Birmingham University, Exeter University, London School of Economics, University of Portsmouth, and the Forest Research. So we have like different working packages. And then the work package 1 is to produce a UK fuel map at the national scale, sorry. And then also like at local level in the Peak District to see like the dynamics of change. Then we also aim to, or we are working on the fuel moisture regime characterization. And then another working packages is like working on the flammability, energy content, and ignability of the fuel map. So there is a team who is like collecting different samples and burning them into a lab, and then saying like what are like the key components of this flammability, assessing the fire behavior. So working package 1 and working package 4 are very like working into collaboration. And then see like the fire security, spatially and temporally over the UK, and then provide like a risk assessment over the UK. So currently at the national level, we are only producing like the fuel maps, and we will assess the fire behavior in different fuel types. But the others, we are just working in local levels, just like some, site studies. So I will explain some of the methods that we are using for building the national fuel map. So we understand when we talk about fuels, it's like the vegetation classes that have similar fire behavior and that they are represented as the leaf and dead biomass that contribute to the spread, intensity and severity of the wildland fire. So what are the key challenges of that? So it is like assessing the reliability of those estimates at the national scale, understanding that the UK has like a complex mosaic, and understanding the role of the different structures when we are talking about fires. So for this, we are generating an AGB map of the UK representative for 2008 to 2018. So this is a static view map. For that, we are also gonna generate a high map over the UK for representative for the same time, and characterize the fuel types according to the AGB variation in the different land cover maps. So here are like the overall methods. So we have for true data, we have national inventory data and some JEDI data for the southern part of the country. We have LiDAR data, which we from DEFRA and Edema Digimap that we use to build a canopy height model. And then we're using like a random forest regression to model both. So with this, we characterize the static fuel map. We are combining SAR and optical data sets, like different like bands and also derive indices. So here is like the AGV data. This is like the information from the National Forest Inventory, from the Forest Research. Unfortunately, and this is one of the challenges that we are having, is like they cannot share the data at plot level. So they are sharing like some aggregate area over woodland areas, but that's why. And also like another challenge is like this is biomass data for woodland areas, but we are also interested in grasslands. So like that's why for us would be better to have like high data from LiDAR, high data from LiDAR. We are complementing somehow with JEDI data for whatever is available in the south of England. And then this is the canopy high coverage at 1 meter resolution, representative for 2016 to 2021. And then this is more LiDAR available from 2010 to 2017 that we are using. This is like the satellite data that I already mentioned. So how we are going to resolve, and this is just like an example, this is not real data. So it's like we are just gonna use, like we are gonna calibrate our model from our final map with the plots, with information at plot level from the Forestry Commission, and then using like a linear regression to calibrate our model, to enhance our model. So the land cover is at 10 meters resolution, and it's provided by the UK Center of Ecology and Hydrology. So we are working with these classes, like to see the different variations, and we are excluding the water bodies as well as the build-up areas. And then, as I mentioned before, the field type is like gonna be built up by this combination of the land cover map and the predicted IGV map. So before, there is like field types from the fire risk at Europe level. And then, but we can see like this is a 1 kilometer resolution, and then there are just 5 classes for the UK that are not really representative. So the idea is like to represent it better. Then finally, I will finish with this. This is like, we were just like looking at the different fire detection per month in different land cover classes, and then we can see like there are like real pattern variations within the 4 countries over the UK. This is still ongoing. We don't have like still results, and then results will use to model moisture regimes, determine flammability and energy content and ignibility across the different fuel types. Thank you.
So, thank you very much. We've got time for 1 or 2 quick questions.
Anna Maria, I mean, from the peak, the major wildfires are heather.
Yeah.
And there's very low biomass. Now, I really doubt your ability to measure heather from JEDI. It just doesn't stand that high. I mean, maybe they don't matter. They can cover large areas, but they don't burn a lot of biomass. That's the first thing. Second thing is that the fuel load isn't just your above ground biomass, it's your dead biomass. And for example, the enormous fires in Yellowstone a few years ago were caused by the buildup of dead wood due to the fact they suppress fire. Now that may not be an issue in the UK, but it may be. So how do you deal with woodland heather fires, moorland fires, and how do you deal with deadwood?
Well, like Jedi is not available over the Peak District, like it's just for the southern part, but that's why we are using LiDAR data at 1 meter resolution, and then yeah, we are like using this. We know that the low biomass is one of the like the challenges in the project, but again, in this like an approximation to build fuel types over the UK, and it would be like better than what it is now. But because we don't have information of deadwood biomass for the whole UK. That was the yeah. Deadwood. Yeah. Yeah, I'm not so sure if I can answer so far this question.
Yeah, thank you very much, Anna. It was really good. Just two quick questions. Sorry. The first one, why do you use JEDI instead of, for example, ISO2, where you will have actually coverage for the whole country? And the second question is, if you could explain a little bit, because you mentioned that you would use the inventory data as a reference and then adjust with linear regression. Can you?
Yeah, like, so the thing is like, the way that the national inventory that the Forest Commission can share the data with us or can help us is like a post-map calibration. So we will send the map and then they will do like a validation, and then we apply like this linear regression that we can recalibrate and like adjust, not calibrate, like adjust the final map to have a better product. Yes, yeah. And the ISA.
You might have to move on though.
Okay.
Sorry.
Okay.
So our next speaker is Casey Ryan.
Right. The clicker. Great. Hello, everyone. Casey Ryan from the University of Edinburgh. I'm not going to talk about that. Where do I get? Oh, there we go. I'm going to talk about a topic very close to my heart, which is estimating woody biomass from radar data. And this talk kind of follows on and is complementary to the talk that João gave just before the break. So I'll skip through some of the background to it because it's kind of part of the same project and same endeavor, but it's a different way of looking at the problem. The first thing to say is that this talk represents a big effort by a lot of people, not just myself, and particularly a lot of people who've collected plot data in the field that makes this analysis possible. You'll see there's about 10,000 plots that go into this analysis. So it's a kind of decade-long effort by about, well, these are just the leaders of the teams, right? So of hundreds of people probably. So in 1992, Tui Latone, who's a sort of pioneer of this work, wrote that there has been an increasing interest in the utility of radar data for forest ecosystem analysis because the intensity in a SAR image at L-band is proportional to the biomass in forest stands. So that sounds good. We can measure forest biomass if we have some L-band observations. But the key question is we don't know the validity domain of the inversion algorithm, which might depend on the SAR parameters, the type of forest, the growth stage, and some environmental variables. So the question is really, okay, these things are related, but how consistently are they related over space and time and ecological type? Our work is all focused on the dry tropics, the savannas and the woodlands and the dry forests that cover about half of the tropics. They're quite understudied. There's very few observational constraints, but the models think there's a whole load of fun stuff going on. So here's a DGVM analysis of the carbon sink, the net biome productivity. You can see in the wet tropics, in the rainforests, it's somewhere about 0.5 to 1 petagram per year. In the dry tropics, it's much more variable, but it's also increasing. And if you believe the representativity of the plot data in the wet tropics, the wet tropical sink is probably declining now. The models think the dry tropical sink is increasing and is probably larger now than the wet tropics. So the rainforests are becoming less of a carbon sink and the savannas are becoming more of a carbon sink. We don't know if that's true. That's a model analysis, and we would like some observational constraints on whether that's real. That's related to my first slide because the dry tropics have exactly the right range of biomass to use L-band radar to detect biomass. And as Israel said, we have good L-band observations, actually going back to the 90s, but really good ones from 2007 onwards. So we have a potential to evaluate whether there is really increasing biomass in the dry tropics over the period that the model thinks that there is. That's our research question. So, 2 questions. How consistent is the radar biomass relationship? If it varies from site to site, and the spoiler is, yes, it does. Why does it vary? And can we understand some of the causes of that variation? There's enough theory there about what radar does and how it interacts with the Earth's surface to suggest that the type of vegetation might be important, the soils might be important. So I'll explore some of those in this analysis. So the in situ data for this analysis comes from the Seesaw network, which is a network now of about 80 people who collect forest inventory data, savanna inventory data across southern and eastern Africa. The red dots show permanent plots where we make long-term measurements. There's about 200 of those in the network. The green hexagons show one-off plots, often done as part of national inventories or similar work by academics and other research groups. We take that plot data set. We use the ALOS mosaics from JAXA. So unlike in Joao's work, we don't have the full poll data over this large extent. We just have the HH and the HV. We would like to have soils data from all our plots, and we do from most of the permanent plots, but we don't from all of the one-off plots shown in green. So we use a modeled soil product from SoilsGrid. We're interested in soil moisture. There were some questions and discussions about that in Joao's work. So here we're using soil moisture estimated from the reanalysis product from ERA land. So that coincides with when the radar acquisition was made. And then I'll use a variety of regression techniques to try and explore some of the patterns in this data. So the first result is here in thick colored lines is each site where we have a data set and the relationship between biomass and backscatter. Not in decibels, just in plain old ordinary units. And you don't need a statistical test to say that there are some differences between the sites, right? So the slopes of those bold lines look different at these 2 sites, for instance, than this cluster of sites. The little thin squiggly line tells you it's just a spline fit through the data. So it tells you maybe whether the data are really linear, which in some cases they are, but in others you can see some saturation, and others you can see complete noise. And then the one that Matt spotted, this site in the Democratic Republic of Congo, there's a clear decline in backscatter as biomass increases. And there's some other studies that have found similar things at high biomass. So result number 1 is that, sure, there are site-to-site effects. And there's been very few studies like this that have compared across sites with the same methods, the same underlying data sets, the same way of estimating biomass at plot level, for example. So radar does different things in different places in terms of biomass. That's the first message. Yeah, the stats are essentially what you can see there, right? That if you put site into your model, you explain a lot more of the variation than if you don't. To try and unpick that a bit, I tried a random forest regression. So now we're not assuming linearity necessarily, and we can look at some of the relative importance of the different aspects of the plot in predicting the backscatter. So variable importance in the random forest, well, the good news is above ground biomass is the most important variable in the regression explaining the backscatter. But the next one is the median diameter of the trees on the plot. So if you're getting your biomass in different ways, either through lots of little trees or a few large trees, you can drive a different backscatter response for the same biomass. The number of stems also drives the backscatter response. This is controlling for biomass. And then the soil variables, soil sand content, that makes sense because we know that radar does different things in sand. The more sandy the soils are. Soil moisture, there are some issues with using the ALOS mosaic to evaluate soil moisture. The coarse fragments in the soil, so this is how many things that are above 2 centimeters in the soil volume. That again makes sense because we think there are some different physical effects in the soil if you have lots of pebbles, and probably that means you have lots of larger rocks as well. And then I'm sorry, something's gone wrong with the slides, but whether you have the ALOS 1 or ALOS 2 satellite also makes a difference. There is a difference between those sensors. If we dig into the random forest regression in a little bit more detail, these are the partial dependence plots. So you can get these out of your random forest regression. They tell you what's the effect of biomass. So this should say sigma naught, right? This is the backscatter. What's the effect of biomass when you hold everything else constant? Or what's the effect of the median density? So now we can look and see what the nature of that relationship is. So the good news is there's a strong relationship between biomass and backscatter. It might be saturating slightly at the upper end, which is what we expect. But when we control for all of those and look at the stem size, if your plot is made up of stems with a larger diameter, then that increases the backscatter up to a certain point where that effect saturates. I don't know if you can see the little ticks, but that shows you where the data, the in situ data lie. So we're not too far out of the range of our data with that result. Equally, if you've made up your biomass with a high stocking density, then you're likely to get more backscatter as well. So there are two ways to get more extra backscatter. You can either have larger stems than average, or you can have more stems than average. And that's quite complicated in net effect because these two are obviously tightly related. The soil sand content, the sandier the soils, the less the backscatter. And that can help to explain some of the site-to-site variation. The others have much more marginal effects than I haven't shown. So if we think about this through the lens of the theory that we often use to interpret radar vegetation interactions, the water cloud model, that exclamation mark used to say sigma naught, sorry. The soil roughness, or all the soil properties, are going to influence the backscatter, but they're mediated by the vegetation, right? The signal has to pass through the vegetation twice. So that's the simple conception of the model. We would like vegetation to be proxied by above ground biomass, but I think the earlier results show that that's not necessarily the case. It may be to do with the full complexity of the elements in the vegetation, not just the biomass. We think the soil signal is probably related to soil roughness and soil moisture, which we can proxy with some of the variables I've used already. So this would be your simplest model. I would suggest the model should look something more like that. I've made it more complicated, sadly. We would like to keep this simple, but AGB is clearly determining the backscatter response. But we know there's a contribution from the stem size, and we know there's a contribution from the number of trees that acts independently of the amount of biomass that is there. If we think about the soil variables, well, that's also complicated because the soil variables will determine the ecology, right? These are not separate things. Different soils give you different tree sizes, different numbers of trees. So there's bound to be some interrelationships between the soil variables and the structure of the woody ecosystem. But of course, we also know that these have a direct physical relationship to backscatter, which is going to be mediated by the vegetation. So this is a mixture now of the radar physics model from the water cloud model. A kind of macroecology model can tell you how these two might be related, the AGB and the number of stems. But then you also need to think about the soil texture. So there's actually a lot of variables in here that might confound each other and might make it hard to interpret what's going on from simple correlational studies like I've just shown you in the first few slides. We do just about have enough data to populate that model. And so it looks like that. Has it survived? Yes, it's just about survived the journey from Edinburgh to this computer. So this is a structural occasion model again, like Joao used. The numbers and the width of the arrows are the standardized effects coefficients. So if you increase your biomass by 1 standard deviation, you increase the backscatter by 0.37 standard deviations. It's a strong relationship there. And that's the same result as I showed you before. And that's the good news. Is that the end? Yeah, okay. This is the last slide. So, there are, of course, these other effects that do add up to something substantial. So the way you get your biomass does matter in comparison to, but is a smaller effect, a secondary effect compared to the total amount of biomass. These are also important, and these can confound some of your analysis if you don't include those as well. That's essentially the point of that one. So why should you care? This is my conclusion slide. I'd very quickly say, you can't think of this system as having scatterers that are all the same. The medium is different from site to site, and your radar model should reflect that. We knew that this could happen theoretically, but I think there's now lots of empirical support for the idea that that's an important effect in retrieving biomass from backscatter. And so when you think about large area mapping, you're going to need to calibrate in different ways in different places. And that has a lot of implications for how you might run a Cal-Val exercise for some of the biomass missions that are in progress. That's it. Sorry, I ran over. Thanks.
Thank you very much, Casey. Have you got any questions?
Thanks for a really interesting presentation there. It certainly got me thinking. So I just was wondering, like, take the picture here that you're showing with the vegetation. Why was it that you just focused on the L-band SAR? Because some of that looks quite low, short vegetation. Have you got any consideration of sort of, are you thinking about using any other frequencies such as C-band, so the Sentinel-1A, 1B perhaps?
Yeah, so we've run this analysis with C-band, the sort of annual minimum C-band, which is less affected by moisture. And you see those same site-to-site effects. They're really strong, and they're correlated to the L-band site-to-site effects. So if you've got a very steep slope at L-band, you've got a steep slope at C-band. But I do think there's more to be done there, because there should be some extra information in the C-band signal that can help us untangle what's going on.
Okay, thank you.
Okay. Yeah, thanks. So the next talk, in the next talk, we're going back to terrestrial laser scanner. It's Phil from UCL. Oh.
Okay, good. Hello. Good afternoon, everybody. My name is Phil Wilkes from UCL. Just say before my talk starts, I've got lots of videos, so they don't think they're going to work, but we'll give it a go. We'll find out. First slide, first video. It does. Great. Okay, so when people give TLS presentations like myself, we usually display these lovely trees like this. You know, there's no background vegetation. They're nicely separated from their neighbors, and there's, you know, even maybe some leaf wood classification. So you get this, you know, great impression that TLS can do anything. But the reality is a bit more like this, where to get that kind of model, there's many hours, as Cecilia will attest to that, you know, manual clipping, removal of background understory vegetation, removal of neighbors, and then there's the leaf wood separation on top of that. So my talk today is about how can we automate this process so that when we scan large areas, we can automate, we can extract all trees from the plot without having to do this manual process. So just going to give a little introduction about LiDAR, terrestrial LiDAR, for those that don't know what it is. Terrestrial LiDAR is an active remote sensing technique, and it builds up these really 3D, very detailed representations of the environment of the trees, in our case, scanning in forests. Our technique, we usually scan 1 hectare plots, and the video on the right, these two are videos here, they're not working. The video on the far left-hand side there, right-hand side to you, sorry, it just shows our technique of building up this plot data. So we scan every 10 meters and build up these, you know, these nice images. And the results are things like this. So as you can see, we've got lots of information. So all the information that we're after, you know, for example, for estimating biomass is encoded in this point cloud data. But we need methods that can quickly or efficiently and accurately extract these individual trees so that we can do the modeling, as Wanshin showed earlier, to estimate biomass. So that's what my talk's about today. So we've, over the last 10 years, we've collected a lot of this data. So we have a big archive to work through. Over 70 hectares so far, mainly in the tropics, but also in temperate forest as well as urban areas. And yeah, a lot in the UK, obviously that's where we're from, a lot around White Woods and things like that. So got a lot of data we need to extract these trees from. Why are we doing this? Motivation is we want to get these plot level estimates of above ground biomass. And the advantage of using TLS is that we're getting this whole tree estimate. So we're not just, not just taking an estimate of DBH, we're including all the crown elements as well. And we're also sampling the whole plot. So we're doing this for every tree in the plot. We're not just taking a selective sample as you might do with a destructive harvest. So this type of information is useful for, cal-val of satellite missions such as JEDI and biomass. And we can also use this information for other interesting projects, and we're looking at change over time. But yeah, so the key is we need to extract these trees. So how do we save hours and hours of clipping on a, on a desktop computer and do this automatically? So over here on this side, we have a flow diagram of how we do this. I'm going to talk through these steps individually in a minute, but basically we have a pre-processing step which sort of standardizes the data that makes it easier to push through this pipeline. We then do a semantic classification, which means we classify wood and leaf components. We then do an instant segmentation. Instant segmentation is extracting individuals from this unordered point cloud. From this, we do that on the wood point cloud. We could then estimate volume and then biomass from this. We can also then add an additional step in which adds in the leaves. So for people who are interested in things like leaf area index, things like per tree. So we are currently running this on Jasmine and also the MagGeo cluster at PML. So yeah, just a shout out for those resources that are very important to our research. So pre-processing, a bit boring, but absolutely essential to, a nice smooth transition through this pipeline of extracting trees. So the key is collecting data in a standardized way. So as I've said, we scan on a 10-meter grid and scan one hectare in this way. And then the filtering and downsampling are also following sort of a standardized protocol. To make this scalable, we tile the data into 10-meter tiles. And that's sort of our processing unit for when we go forward. The next step is the semantic classification. So this is using a deep learning method developed by a PhD student at the University of Tasmania, Sean Krasansky. This is a new method, and I think this was a real game changer in this type of analysis. So we used to do the wood leaf separation step after we've extracted the individual trees, but doing it beforehand allows us to, it sort of increases the gap between the trees. So doing this extraction doing the extraction sort of separates the trees out, makes it much easier. As you can see here, we've got a wood point cloud. So the next step is the instant segmentation. I'm going to skip over these slides quite quickly. If we want to talk about it in more detail, we can do that afterwards. So we've got our point cloud. It's been classified, but it's still an unordered point cloud. There's no individual trees identified in that. So that's what the instant segmentation does. This is how we start. We're working on our 10-meter grids so that we can sort of parallelize these processes and run it much quicker. So we have to buffer the point clouds because these trees don't fit nice into our 10-meter grid. So they, spill over the sides. So we use a buffer in the tropical forest. This buffer can be up to 70 meters because the trees sort of lean over and they've got huge crowns. Next slide. There we go. But for this instant segmentation, we're just using the wood point cloud. Here it is for side-on view. We do some clustering because, sorry, I should have said this instant segmentation is we build a graph to do the instant segmentation. So we have to find some nodes. We do some clustering. We find the bases of the trees as the basis of this graph analysis. And then we use the Dijkstra shortest path techniques, which I'm sure some of you are aware of. This figure on the right, your right, is slightly misleading in the way that we construct the graph. So the edge weights for the graph in previous studies have sort of calculated the path length, so the distance from the base of the tree to the top of the tree. But this can cause errors when if you've got tree crowns growing into each other. So we've developed a new technique which basically uses convex holes to around each of these clusters from the previous slide, and then the distance between those clusters is our edge weight in when we build the graph. So it's more like of a path error instead of a path total length, and this proved more accurate in extracting the trees. So we run this with this graph analysis, and we can pull out our individual trees here, colored in different colors. Here's a top-down view. We can add the leaves on. The leaf adding on method is very similar to the other method. So we basically, instead of clusters, we put voxels around the leaf points and then use a similar graph-based method to add the leaves back on. And then we attribute each stem to its 10-meter grid. So this is the so it's sort of germination point. If it's germinated within inside the 10-meter grid, it's attributed to that one. Okay, so we've got our individual trees extracted. We can run that across whole plots. Great. Now we use a similar method to what Wan Xin was describing earlier, tree QSM. So this allows us to, so tree QSM basically encloses the point cloud in a set of cylinders, topologically coherent cylinders, so that we can then pull out attributes such as branch length or total surface area. But the one that we're most interested in is total volume. We multiply that by an estimate of wood density, and that gives us above ground biomass for each tree. So on to results. So the tree I showed you at the beginning that I said I hand clipped, I lied a little bit. It was one part of our automatic classification. So we're sort of really pleased with these results and how well it performs even in dense tropical forests. So the real key is a figure like this. So these are all the trees, over 500 trees from dense tropical forest plot. We've automatically extracted all the trees. So this kind of work would have usually taken months, and we can now do it in a week, 2 weeks at most. And the other benefit is that it's already leaf-wood classified, so we can remove the leaves nice and easily. So we can start fitting our tree QSM models to this and estimating biomass at a whole plot level. Another plot, this one's in Gabon. Cecilia's recently been there. Again, this is even more trees, slightly different forest structure, but again, a couple of weeks, and we've got all these tree models, which is fantastic. This is forest in Australia. So this was Kim Calder's sort of the first TLS destructive harvest estimates. And again, works really nicely, very happy with the results. But how do we know how good they are? That's the big question. So on this figure, along the y-axis is, along the x-axis is hand measured trees, hand clipped trees, and on the y-axis are automatically segmented trees. We just got a few attributes here that we're comparing. As you can see, it performs pretty well. We're pretty happy with these results. So I'm gonna have to quickly go through these slides. So unlocking our data, unlocking our archive. So these are we haven't gone through the whole thing yet. We've done about 12 hectares and we've already extracted 5,000 trees. And we can look at, attributes of these trees, looking at diameters and total volume. So we've got some really big trees in our data set. This one's over 70 tons. And we can start to, interrogate this database. And we can see how the biomass is distributed within the plot. So really, this is early analysis. We need to look into this slightly more, but you can see that the trees in red are the trees that contain 50% of the total plot biomass. Trees in grey are the rest of the trees. So for example, for some plots, it's 4 or 5, 10% of the total population of trees contain 50% of the biomass. So we can do this for all our trees across our 70 hectares, and that's the key. Second to last slide. So this is a comparison with the Jerome Charve database, where they've done a lot of destructive harvest. And as you can see, so this is our distribution of DBH, and their distribution of DBH is similar. Same with height, but our estimates of biomass are very different. We're estimating a lot more trees have we have more trees with less biomass compared to the Charve database, even though we have a similar DBH and height distribution. So that's my last slide, apart from this. Oh no, sorry, limitations, conclusion slide. I'll whip through this one. So the limitations are the semantic segmentation model is quite limited. It's worked really well in tropical forests, but some savanna sites hasn't worked quite so well in. I'll go to future directions. We want to process the rest of the archive. We want to make it open access. So we need some guidance, and probably the best way of doing that. And improved tree QSM fitting, so referring to Wanshin's work there. This is my last slide, I promise. Another plug for Silver Laser, and if you guys like watching trees spin around slowly, then be sure to follow TLS Trees. Thank you very much. Hi. Yeah, that's a good question. That's something we haven't really tackled yet, because wood density can be very variable even within a tree, even within different components of the tree. So at the moment we are assuming a wood density, uniform wood density across species. But I mean, even for the Sharv equation, 24 Sharv equation, there's a wood density parameter in there. So the short of it is we don't have wood density for all these trees, so we have to make an assumption. Yeah, so Wanshin presented some of Andy Burt's work, and they did do some wood density over height measurements. Yeah, so that's in Andy's paper.
Well, oh,
hey, hi.
Thanks for that. That was really great. The last slide with the difference in biomass and the Sharv versus your estimate, I was like, whoa, okay, does that mean there's half as much biomass in the tropics as we thought before the talk? That seems quite a big difference.
Yeah, it does. I should say that we only have 5 plots in this, and I think it's very much influenced by one of the plots that we have in Malaysia, and it has a lot of much smaller trees in it. So, and that probably skewing our database at the moment. So this is definitely a first cut of this. So, but generally, yes, I mean, does it mean that the Sharv database, which one is representative? I guess that's the question.
I can but maybe just comment. There's so many assumptions that go into the above ground biomass, including things like density and so on. You're better off looking at the height and the DBH and the differences that occur there. So that's why we compare typically at volume level rather than AGB level, and then looking at the heights and so on. Then that gives, because we trust those a lot more, because you don't have to make those assumptions.
Does this mean at some point we've got to scrap the Sharv allometry?
Because we need more trees in it, because it's very unrepresentative of trees more generally. And then, there are other ways to do it. I wouldn't say you'd scrap it, but you'd add to it along the way and have different ways of doing it.
Yeah, so this would be a complementary database to Sharv's. Cool, thanks.